So it's an honor to be here. This is my third time giving a lecture before this audience, and I want to thank my hosts for inviting me. I'm always happy to come back. Mine? Is this, everything's fine. So, okay. So the topic I want to discuss today is it's something that is very basic. It's a very basic question in biology. It's a very basic question in the history of our planet and the history of life. And yet it's something that is rarely found in textbooks, and it's something that people don't really work on as a scientific subject of research. And this is a subject which I've discovered recently, and it's actually something which we have spent a lot of time in recent years trying to come to grips with. So I'm going to share with you first some of the broad background. So because it's a general audience, I'm going to present what the problem is and what the big mysteries are. And then in the last part of the lecture, I'm just going to give you a flavor of how we work on it. It's something which is a technical part of the lecture, but I think it's nice to share with the audience how we actually do our work, how the science actually proceeds. So you'll notice that the title is slightly different than in the pamphlet. So the pamphlet is called The Evolution of Cells. And I'm going to restrict the subject a little bit. It's called The Evolution of Compartmentalized Cells. And I'll explain what this means. So this painting is something I came across a few years ago. It's a work by an Aboriginal artist from Australia, whose name is Shorty Changarai, who's passed away now. But he was a leader in Aboriginal art movement, and these paintings actually have a lot of meaning. Apparently, the way you're supposed to read an Aboriginal art painting is there's a certain code, and if you actually know the code, this entire painting tells a story. You know where to start, where to stop, and all the places in between. And of course, I don't know the code, and nobody who's not an Aboriginal knows the code. But when I saw this painting, it reminded me of something that looks very biological. In fact, these things here reminded me a lot of cells. And so I just kept this painting on my computer and I kept looking at it. And I still think it's a very beautiful painting, but it also sets, I think it gives me a nice background to set the theme for the rest of the talk. So here's how I see it. Okay, so these are how I see cells and so on. But here, if you look at it, it looks a bit like stars and galaxies and things like that. So what this painting does for me is it brings together these two scales. It brings together the scale of the universe and the galaxies and the stars, and brings together the scale of cells and molecules and proteins. Right? So this is exactly what science is all about. And why are these two scales important? It's important for the following reason. So imagine this is the timeline for our universe. Okay? How old is the universe? So we know by now from cosmology and various kinds of estimations that the universe is about 14 billion years old. 14 billion. A billion years is 1,000 million years. So it's a 14,000 million years old. That's the origin of the universe 14 billion years ago with the Big Bang. And very soon after that, you start to get the collapse of matter. You start to get the first stars, the first nuclear reactions starting, and the formation of galaxies, formation of structure in the universe. That's very, very quick. Not long after that, so 5 billion years ago, you have the dust cloud around the sun collapsing, and then you have the solar system forming. You have the Earth forming. So the Earth is roughly 5 billion years old, same as the solar system. Now when the Earth first forms, it forms at a high temperature. Right? It forms in a cloud of very high temperature gas and dust. And therefore it's almost molten. It's complete, it's like magma, lava, it's a complete molten surface. And the Earth slowly cools and solidifies, and 3.5 billion years ago is when the rocks first become solid. Okay? So imagine, that's the first time a rock can actually be formed. And we now have, if you look at the rocks that you dig up at deep strata and the surface of the Earth, you can date them using various techniques, and you find that they date to about 3.5 billion years ago. So the amazing thing is that in these rocks, which are the first rocks to form, you find fossils. Okay? So this is an amazing thing. There are fossils in the earliest rocks that there are on it. That means that life arises on Earth very, very early. Almost as soon as it becomes possible to have life, you have life. Okay, so these are the first cells. Now this is a problem I'm not going to talk about. So I'm not going to discuss how life first arises. The reason is because I don't know anything about it. In fact, nobody knows anything about it. Right? It's a very difficult topic, a topic about which there's a lot of speculation, but there's very little hard science. So that's 3.5 billion years ago. The next big thing that happens on Earth is again a huge event. You know, these days we talk about climate change. Why do we have climate change? We have climate change because there's a huge amount of carbon dioxide that's suddenly being released into the Earth's atmosphere. It's changing the temperature. So this kind of event actually happened 2.5 billion years ago. How did it happen? There were bacteria on the surface of the Earth. They cover the Earth. Life is invented. There's chemistry going on everywhere. And one group of bacteria, which are called cyanobacteria, invent the process of photosynthesis. Okay? Before that, there is no photosynthesis. Once you have photosynthesis, what do? They use solar energy and they fix carbon, they create sugars. Right? Now the byproduct of photosynthesis, as you all know, is oxygen. So what happens when photosynthesis is invented is oxygen gets released into the atmosphere. Okay? So before that time, life on Earth does not require oxygen in the atmosphere. It's anaerobic. After this time, oxygen is completely pervading the atmosphere, and therefore life becomes aerobic. And this is the starting point of my story. Because you and I, every cell in our bodies, we need oxygen. And the cells in our body are a fundamentally new type of cell, which arise 2.5 billion years ago in order to take advantage of the oxygen. Okay, so these cells are called eukaryotic cells. You've heard about it in your textbooks. So my entire lecture is going to be about how these cells first arise, okay, and how they evolve in time, eventually leading to animals like this. So this strange abbreviation here, LECA, I'll tell you what it is later, but the expansion stands for the eukaryotic common ancestor, the last eukaryotic common ancestor. Okay? So this is, if you take all the eukaryotic cells on Earth, I'll tell you what I mean by that, and you trace them back until they reach a common point. When was that point? That point is 1.5 billion years ago. Still a long, long time scale. And finally, 700 million years ago, you get the first animals and so on and so on and so on until you get a human being. So it's a very, very, very long timeline and a very long time for life to have evolved. My part of the story runs there. So that's the history of life on Earth. Now I'm going to tell you the history of cell biology, the history of the science of cells. So Paul Nurse, who's a Nobel Prize-winning biologist, he likes to talk about the great ideas of biology. Now everybody knows one of the great ideas of biology is the theory of evolution, Charles Darwin. It's one of the great ideas of biology. But in Paul Nurse's opinion, and I agree with him, the other great idea of biology is the cell theory. Okay, so what is the cell theory? So in the 1600s, there was a fabulous new invention, the microscope. Right? You put a couple of lenses together and suddenly you can look inside tissue samples, you can look inside water, you can see all kinds of structures there that you could never see before with the naked eye. And two discoveries, both made by microscopes but by very different people in very different contexts. So you have Van Leeuwenhoek and you have Robert Hooke. Van Leeuwenhoek was just looking inside water. He would just take samples of water from ponds and look inside, and he would see thousands and thousands of small organisms swimming around, and he called them animalcules. Okay? He called them animalcules because they reminded him of big animals moving around. They had structures that looked like legs, structures that looked like tails, and so on. But these creatures were not animals. They were single cells. They were free living cells that were swimming around in the water. So he publishes his books and he draws images of these cells and so on. So he's identified this completely new realm of life on Earth. Nobody even knew it existed. You know, all people knew before that was plants and animals. And suddenly you have these creatures. At the same time, Robert Hooke is examining cork, like you put in wine bottles. Right? He's examining bark from trees and so on. He puts those under a microscope and he sees, I don't know if you can see the picture, but he sees that what looks like a big tissue actually breaks up into small little units. And he calls them cells because it reminds him of the cells of a prison. You know, you have cellular jail because it's got these cells, and that's where the word cell comes from, because it looks like little units. Okay? So both these men, roughly 10-15 years apart, discover cells. And over the next 100 years, people come to realize this very important thing. All life on Earth, whether it's big objects like us, whether it's trees, humans, blue whales, or free living organisms, all life on Earth is made from cells. Okay? That is a scientific discovery of the greatest importance, because it means that in order to understand humans, in principle, you don't have to study humans. You can study the cells of fruit flies, or you can study the cells of bacteria if you want to, because they're all cells, and cells have a lot of things in common. Okay, so this is the first big idea. And this idea has a lot of medical importance, because from the cell theory came the idea that diseases were caused by germs that were invisible but they were present in our surroundings, and that's how ideas of hygiene, cleanliness, and so on came about. Okay, so this cell theory actually saved a lot of lives in the centuries following these discoveries. So it's very, very important. But I'm going to skip all that, okay? I'm going to jump hundreds of years to very recent times, early 1900s, let's say 100 years ago. 100 years ago, if you asked, 100 years ago we already knew special theory of relativity, so we're fairly well advanced in scientific terms. But biology is still lagging behind a lot. If you asked scientists, biologists, what the big dichotomies are in biology, what are the different types of creatures, what are the different types of organisms, they'll say that's very easy. There are unicellular, free-living cells, okay, like Leeuwenhoek studied, and there are big multicellular organisms, like plants and animals, and these are the big differences. Okay? That's how you classify life. It's unicellular and it's small, or it's multicellular and it's big. And they were wrong. Okay, those aren't the big dichotomies in biology. So let me explain what I mean. So remember, in the 1900s, the microscopes were not very much better than they were in the 1650s. There was no new technology, which is better lenses. But in the 1940s, something new happens. A new type of microscope. We all know what that is. It's the electron microscope. Once people got their hands on the electron microscope, there was a period of intense study. Anything they could get their hands on, they would just put under the microscope to see. Right? This was how they were working. They would just go into their backyards, they would cut some tissue, put it under the microscope to see. And that's when people started to realize that there's a distinction that they were not aware of before. It's not that there are free living cells and multicellular organisms. It's not that. There are actually two types of cells which are called prokaryotes and eukaryotes. Okay, you've read this in your textbooks, even I read it in my textbook. I didn't realize what they were talking about. So we know what they mean now. Prokaryotes are like bacteria. They're small, they're about 1 micron big. 1 micron is the wavelength of light. Eukaryotic cells are like our cells or plant cells. But there are also many free-living eukaryotes. Right? All the creatures that Van Leeuwenhoek saw were free-living eukaryotic cells. They are a more complicated type of cell. And if you put them under an electron microscope, and I'll show you some images later, and you slice them through, you'll see that these cells are much bigger. They're 10 to 50 microns in size. And inside the cell, like you see in all your textbooks, you see all kinds of little organelles, you see traffic systems, transport pathways. So these cells are not just simple bags. Eukaryotic cell is almost like a city. It's like a city with different districts, and there's one district which is a factory where you make the goods, and then there are roads on which you ship things from one side of the cell to the other. There are places where you destroy things. Right? So a eukaryotic cell is a very, very complicated organism. And it was only discovered by electron microscopy. So very good. So in the 1940s we knew there are two types of cells, simple cells, bacteria, and complex cells, like human cells, like amoeba, like plant cells. Okay? How did these two types of cells come about? And this is what I'm going to tell you about. Ancestrally, originally on Earth, there must have been one type of cell. So how does a new type of cell arise from them? Okay? So this lady is a very important figure in the history of cell biology. Her name is Lynn Margulis, and she passed away last year. Lynn Margulis was actually married to Carl Sagan, the astronomer who was science popularizer. He did the TV show Cosmos, which they showed on Doordarshan actually in the 80s. So Lynn Margulis had an idea. So she looked at these eukaryotic cells, and she looked at all the little all the organelles that were inside eukaryotic cells. And in particular, she was interested in one organelle, which is called the mitochondrion. Again, it's something that you hear about in the textbooks. The mitochondrion is inside every cell in our bodies. Okay? It's a small rod-shaped structure. And Lynn Margulis looked at it, and it just occurred to her that these little pieces look like bacteria. They just look to her, they're reminiscent of bacteria. So she had an idea that maybe these complicated cells arose in a very strange event where two individual cells fused, okay, to make one more complicated organism. So that was called the endosymbiont hypothesis, and I'll go into a little bit further. So in fact, Lynn Margulis was not the first person to do this. There was a Russian scientist, Merezhkovsky, who proposed the same thing in 1905. I'll show you that data. But this becomes very important because this is the beginning of the time when the evolution of cells becomes a scientifically acceptable field of study. Okay? You can make a hypothesis, you can test the hypothesis. Fine. Another important piece happens in 1977. Carl Woese, who you see over here, he is another pioneer. So he did what might be described as the first, what you might call large-scale genetic analysis. Okay, so what he does is he just collects samples of RNA. So RNA is a molecule that exists in every one of our bodies as part of ribosomes, every cell on Earth, in fact. And so he collects RNA from different cell types, and he just classifies them. And he realizes that, again, we were wrong. There are not two types of creatures, not prokaryotes and eukaryotes, but there are actually three great kingdoms of life. Okay, these three great kingdoms are called bacteria, archaea, and eukaryota. Bacteria and archaea are both prokaryotes. They are simple cells with no internal structure. The eukaryota are the eukaryotes. Three great kingdoms of life. Okay, now we come to the present day. In the present day, we don't have the very sparse amount of data Carl Woese had in the 1970s. We have access to thousands of genomes. We have access to cell biology. We have access to techniques of protein chemistry, and so on and so on and so on, where we can again revisit the question of eukaryotic evolution and see whether we can say something. Okay, so that's enough of the textbook stuff. Let me just show you some amazing pictures. Remember I said the oldest fossils in the rocks? These are the fossils. Okay? So you can find these fossils in certain very deep strata, particularly in China and in Australia. There are some very deep beds where you can collect samples. You can imagine it's a very, very difficult thing to do. These fossils are microns in size. Microns. So if you're a scientist working in this area, you have to keep digging up rocks, slice them finely, and keep looking on a micron scale until you stumble onto a fossil. Right? It's much more difficult than coming across a dinosaur bone. Right? So it's a very, very difficult feat. But there it is, and you can date the rocks that surround them. That's how we get this number. GA means giga years ago, billions of years ago. So this is 3.5 billions of years ago. Now let's look at some slightly more recent fossils. These guys are again found from rocks in China. And if you look at these structures, they don't just look like simple rods. Okay? They look much more interesting. They look like balloons. And they have all kinds of things sticking out of them. They have structures inside. They have things on their surface. Right? They have a rigidity. Right? So this is the first evidence of complex cells. Not simple prokaryotes, but complex eukaryotes. And how old are they? They are 1.8 billion years old. Very good. So now you can take these fossils and you can take them to a cell biologist and you can say, what type of creature is this? Is this an alga? Is this an amoeba? Right? Is this a protist? And if you look at these cells, they don't look like any creature that is alive today. Okay, these are strange fossils. They are definitely cells, but they don't belong to any of the families of organisms that are alive today. So this chart shows you the origin of modern types of cells. So again, we're going from 1.8 billion years ago to 850 million years ago, 560 million years ago to the present. Okay? These bar graphs, if you look at it, each color in the bar graph represents a different type of cell. These are all different types of cells. Okay? Let's see. These represent some of the more modern cell types. These represent some of the ancient cell types. And as you go forward in time, you find more and more types of cells emerging. Except during... very good question. So it's a very tricky subject. There's always a lot of let's say subjectivity to it, but you can do a couple of things. First of all, you can trace, you can do chemical traces. You find traces of phosphate and other kinds of things which are characteristic of biology. Okay, there are no lipids, but you only find the inorganics left. Secondly, it's very difficult to imagine any, let's say, non-living organic process depositing things that look like this. So it's a subjective call, but they look, let us say, very much like cells to our eyes. Right? And this is not just at this scale. You can slice them much more finely under the EM, and you see much finer structures and so on. So it's a very good question. The subject is definitely not without controversy. So let me tell you that these things are still controversial. People still object to say whether they are cells or not, because they're much older, and the chemical evidence is gone. But these things, especially in more recent times, it's fairly well agreed. So what happens recently, coming up to the present day, you find that from the original ancestral complex cell, whatever it must have been, there's a huge diversity of cells on Earth now. Okay? Now from Darwin's day, we've known there's been a huge diversity of animals and plants. Okay? This is what Darwin based his theory on. How do you have such a diversity of plants and animals? And so we all know the different types of animals and plants. But what is less well known is the diversity of single-celled organisms. But we now know that today. So I'm going to give you a little example of all these types. So here I have examples of different types of cells from many different parts of what's called the eukaryotic tree of life. What is a tree of life? Just like Darwin drew, it is something called a phylogenetic tree. It just shows the relationships between different organisms. Right? Two organisms that are more closely related have a more recent branch. Two organisms that are distantly related have a much older branch. Okay, this is a phylogenetic tree. And you see that from the origin of eukaryotes, there's been a big diversification. Many different forms have emerged. On this tree I've drawn in particular two very important events. In orange I've drawn what I described was Lynn Margulis's work, the mitochondrial endosymbiosis, of which I will speak about a lot. In green is the other major endosymbiotic event, which is chloroplasts. Okay, remember I said bacteria invented photosynthesis. But how did plants become photosynthetic? They didn't reinvent it. They just took up the bacteria. Okay? So that's another endosymbiotic event that happened. It's not that rare. Okay? So what are the different types of organisms that we'll consider? Well, in this tiny branch of the tree are what are called opisthokonts. Okay? It's a strange name. But in the opisthokonts are the animals, in particular humans, fungi, certain other creatures. Here are the amoeba that people like to talk about in the textbooks. Here are the plants and green algae and all the extant photosynthetic organisms. Here are the alveolates, in particular ciliates, again paramecium, which is in the textbooks, is up here. Rhizaria, in here we have what are called diatoms, the sort of diversity of creatures that live in marine surfaces. They make plankton and so on. So here is the diversity. Now you might imagine that if eukaryotic cells are so diverse, we can't study how they evolved. Right? Because they're all different. It's very hard to make connections between them. But this is where the big mystery comes. So here is the most surprising fact. All these cells, they look very, very, very different from each other. But all of them fundamentally are the same. What do they look like? They look like this. This is why this is the picture that's in the textbooks. Because it is completely accurate. Every eukaryotic cell, all the cells in your body, amoeba, diatoms, plants, have more or less these features. Okay? And I'm going to go over them carefully because it's important. The cells are called eukaryotes because they have a nucleus. So karya, the word derives from nucleus. This is the nucleus. The nucleus is where the DNA is packaged. Okay? And when I learned this in school, I thought the only difference between prokaryotes and eukaryotes is that one has a nucleus, the other doesn't have a nucleus. But that's not true. Eukaryotic cells have a nucleus, but also there are dozens of other features that they share that bacteria don't have. What are these features? They have these orange things. Those are the mitochondria. Okay? They have this funny dotted structure, which is called the endoplasmic reticulum. That's where proteins are synthesized. Okay? They have lysosomes, peroxisomes, Golgi body. Okay? Various kinds of internal organelles where different kinds of chemistry happen. Okay? They have these things. These are called flagella. There are some cells in our body that have flagella. Free living eukaryotes have flagella. This is how they move around. Okay? They have bendable membranes. Right? And they have these things called actin and tubulin, microtubules. These are tracks. They're literally the highways along which material flows inside the cell. Okay? So you see, The interesting thing, the eukaryote is not defined just by the nucleus. It's defined by every one of these features. Okay? So you have to ask, when you do archaeology and you're looking at human evolution, in fact today in the papers I read, there's some kind of intermediate fossil between snakes and lizards. Right? If you read the paper today, there was a story about that. In the same way, when you look at human evolution, you always look for intermediate forms, right, that are partially share the features of apes and partially share the features of humans, and that's how you reconstruct evolution. Right? But in this case, between eukaryotes and prokaryotes, there are no intermediate forms. One side has everything, the other side has nothing. It's as if you started off with a fish and suddenly you ended up with a human, full legs, brain, everything, right? With nothing in the middle. So that's the big mystery. So again, the same picture. This is the timeline, 3.5 billion years ago to the present. Here are the three great kingdoms of life: bacteria, archaea, and eukarya. These two are prokaryotes, these are eukaryotes. Okay? What do these abbreviations mean? LUCA is the last universal common ancestor. It's the last common ancestor of all living things. LECA is the last eukaryotic common ancestor. The last common ancestor of all eukaryotes. Now somewhere about 2.5 billion years ago, remember before 2.5 billion years ago there was no oxygen. Okay? Mitochondria need oxygen. There's no point having mitochondria if you don't have oxygen. So there's no eukaryotes before that time. Somewhere around here, some cell decided to go its own way and eventually leading to the eukaryotic branch. Okay? 2.5 billion years ago. But if you remember, the ancestor of eukaryotes according to the fossil record dates back to 1.8 billion years ago. So there's a very long branch on the range of 500 million or 1 billion years, okay, along which a huge amount of stuff is going on. You start off with a simple cell, suddenly you have mitochondria, nucleus, cytoskeleton, bendable membrane, ER, so on and so on and so on, until you get here. Okay? So at some point in time there must have been a creature that, say, had a nucleus but didn't have mitochondria. Or maybe had a mitochondrion but didn't have cytoskeleton, right? Those creatures must have existed. But unfortunately for us, they've all died out. They've all gone extinct. They didn't survive till the present day. The missing links are truly missing, right? Except in the fossil record, which is very difficult to interpret. So, how can we possibly study what happened along that branch? And this is the big mystery. We want to know what series of events happened here that led to the creation of complex cells. We know at least two of the pieces. One is the mitochondrial endosymbiosis. The other is the endosymbiosis of chloroplasts to make photosynthetic organisms. But other than that, we know nothing. So I'm going to describe to you over the next few slides some hypotheses people have put forward. Okay? Let's start with one of the oldest. This is Merezhkovsky. This is the Russian scientist I told you about. So he had this strange idea. So Darwin, in his notes, there's a famous picture where he draws a tree and he says, I think, and he draws evolution by descent and branching. Okay, so since Darwin, biologists have been trained to think that life proceeds by descent with modification, right, on a tree. Similarly, here is a tree. That is the ancestor, these are all the descendants, right, and they keep on differentiating. Merezhkovsky had this very strange idea. Maybe there are separate branches of the tree and they can connect up in funny ways. This is not a tree, this is something very strange. Okay? So he proposed the idea that you could have hybridization between different parts of a tree. Now he wasn't doing this very seriously, the data were not well supported and so on, but he put the idea out there. Fast forward to the present. I'm going to show you a figure from a paper that's published in 2011. Okay, this is what life people think looks like now. Okay, this is from genetic data. This is from DNA and protein sequence data. Here is the last universal common ancestor. On this branch, we have bacteria, right? Actinobacteria, bacilli, so on and so forth. On this side, we have the archaea, which is another very mysterious kingdom. They're still single-celled organisms, but they're very different in their chemistry and in some sense their lipid structures and so on. Okay, so you have bacteria, you have archaea. Here you have the eukaryotes, animals, fungi, plants and so on. Right? You do the protein analysis from this and you don't find a tree. You don't find that these guys branched off, then something branched from here. You find something very strange. You find the two great branches like this, and suddenly, in a very mysterious event, a lot of signal comes from here and from here to create eukaryotes. Almost a magical event where two great kingdoms, some cells come together to generate the cells that then eventually make our bodies. Right? Still very controversial. Still very controversial. But this is DNA data. This is not somebody's scribblings on a pad. Okay? So, fine. This is something you can publish, but what does it mean in terms of cells? Unfortunately, there are as many ideas about what this means as there are people working in the area. Okay? So this is just a limited subset. I'm going to show you quite a few more. I don't know if you can see this. Is it possible to turn the lights? Yeah? For the next couple of slides, you need to, and then we can turn it back on. Yeah, that yellow is a bit. I think that light is a. Okay, I see. Okay, fine. Okay, so is this visible? Yeah. Okay, good. Thank you. Okay, so here's what we're going to do, right? Okay, it's still visible. Thank you. So here's what we're going to do. So remember, there's this funny, strange signal. Bacteria, archaea, some kind of combination leads to eukaryotes. What is this combination? Is it mitochondria? You know what? Where does this come from? So here are all the ideas. Remember Merezkowski, early 1900s. So he has an idea that the reason cells have internal compartments is very simple. One cell just grabbed another cell, just engulfed it, and then that other cell just lives inside it as the nucleus, and similarly as chloroplast and so on and so on. That is a basic sketchy idea. What are these cells? Are these bacteria? Are they archaea? He doesn't say. But there's a big problem with this. Okay? The problem is that any cell that is sophisticated enough to make pseudopodia, okay, already has actin, cytoskeleton, motor proteins, cholesterol. In other words, it looks like a modern eukaryote. So where did that come from? We don't know. Okay? You can keep on going. I'm going to skip this one. Very specific hypothesis. Lynn Margulis, 1970s. So she has two ideas. One of them is that a bacterium, a free-living aerobic bacterium, gets swallowed up by the cell. This cell has bendable membranes. This cell is just a bacterium. So that blue guy becomes the mitochondria. Right? Continues. This green one is the photosynthetic bacteria which become the chloroplast. But then Lynn Margulis goes a little wild. Now she starts to think that anything about the cell comes from fusion of cells. So for example, she thought until the end of her life that these flagella were actually free-living bacteria that got taken up by cells. Now that turns out not to be true. Okay, so you can't explain everything by hybridization. Who is still alive, he's a grand old man of cell biology, Nobel Prize winner. He comes up with a very strong statement that if you have a big cell and that cell has lots of organelles inside, most of those organelles arise by endosymbiosis, by one big cell engulfing another little cell. This is very dangerous, okay, because a cell has a lot of organelles, but the mitochondria and the chloroplast are special. I don't know if you know why. If anybody has any idea. The mitochondria and the chloroplast have their own DNA. Okay? They have their own DNA, which looks like bacterial DNA. They have their own ribosomes, everything. Okay? Which is a strong indication they used to live on their own. No other organelles in the cell have their own DNA. So why should they come from the outside? Other very similar ideas. Okay, we can skip. Here's now where people in the 80s. Why the 80s? Because here is when why am I missing this? This is where the archaea, Carl Woese, yes, discovers archaea, bacteria, and eukaryotes, right? So only post the early 1970s do people realize there are three kingdoms, right? And that strange signal of eukaryotes contains some signal from archaea and some from bacteria. So now people start to really make cellular claims, right? So, for example, Cavalier-Smith says that a regular bacterium, for example, E. coli, that's a regular bacterium, and an archaeobacterium somehow fuse to give the modern day eukaryote. Okay, but how this happens, that's very vague. There are more very, very specific recommendations. You have a bacterium, an archaeum, this one swallows that one, but somehow all the archaeal stuff is left out. Okay? It's very peculiar. In our bodies, certain types of proteins come from bacteria, certain other types of proteins come from archaea. Right? The lipids come from bacteria, so how do you pick up some pieces and not others? Okay? You can go on making various kinds of claims, and everybody has a different opinion. What comes outside, what comes inside. Okay? So, I'm not going to bore you with this, except to leave you with one interesting piece over here. This is a person named John First, who works in Australia. He works with a very interesting bacterium, which is called Gemata. This bacterium itself has internal structures. So he thinks that this represents the ancestor of eukaryotes, or at least represents a cousin of the ancestor of eukaryotes. And that thing just diversifies to become a modern type of cell without mitochondria. And so on and so on and so on. So I'm not going to continue on this line, but just to give you the impression that the modern day eukaryote, there are many theories as to how it happened. What is the difference between all these theories? The difference is difference of opinion between different scientists. What are the types of cells that came? Everybody agrees that different types of cells came together to make eukaryotes. Which types of cells they were, they disagree. They also disagree on the order of events. Right? Did we have cells with a nucleus first, then the mitochondria went in? Did the mitochondria come first, then a nucleus was formed? Right? These kinds of things are still very, very controversial. Okay? So I'm going to leave behind, in the last, let's say, 5 minutes, I'm going to zoom through some of the work that we do to address some of these questions. Okay? So, coming from a modern cell biology institute, you address many questions in biology by looking at molecules. Okay, you don't look at morphology, you don't look at taxonomy, you look at molecules. So then it becomes important to ask, here are the cells, here are all the structures inside cells, nucleus, ER, mitochondria and so on. Right? That is a cell, let's say, morphological way of looking at it. But we can ask the parallel question, what are the proteins that are responsible for maintaining all these structures? Right? So here's a list of all these proteins. Dynamin, escorts, GTPases, snares, so on, the RADs, and so on and so on and so on. I'll tell you what few of these are later. Okay? So we can track the history of these proteins because we're very good at that. I mean, what we can do is do molecular biology and phylogenetic reconstruction. This is a way to find the ancestry of proteins. It's a very well-developed quantitative subject. Okay? So rather than tracing the history of the cells, let's trace the history of the molecules. That's the idea. What are these molecules? Very easy. So if you have, this is supposed to be a big cell, and that's the cell's external plasma membrane. That's the nucleus, that's the endoplasmic reticulum, Golgi, and other organelles. To maintain this membrane system, you need two fundamental processes, right? You just need to move things from one membrane to the other. How do you move something from one membrane to the other? You take the first membrane, and you simply make a bud. It's called a vesicle. You load it with cargo, you move it on a track, literally like a highway, and then you deposit it in the destination compartment. Okay? So to make a cell, you need two kinds of processes, vesicle budding, vesicle fusion. And the molecules that are responsible for this, for vesicle budding, are called RAD GTPases and coat complexes that make the vesicle. The fusion is mediated by other proteins, again some RADs and snares, which actually connect to lipid surfaces and bring them together. So you can make a vesicle, move cargo, deposit it. If you can do just that, then you can couple all the different membranes in the cell. You can make organelles. Okay? So the general idea is the following. If that's an organelle, and that organelle is defined by these particular molecules, in this instance this RAD and this syntaxin. I won't get into the details of what they are. Very general idea is, when you had the original cell, it had maybe just one internal compartment. Our cells have dozens of compartments. How do you get so many compartments? Easy. You keep making new genes, and by making new genes, which is very easy, you can make new compartments. Okay? So that's the idea. You take the eukaryote, which already has an internal compartment, and then you keep on adding more compartments. This is what I call organellar diversification. Okay? So now this is the technical part of the talk for the next couple of slides. So I don't want to get into it very deeply because I know there's a broad audience over here, but I'm going to present the material anyway. And if anybody wants to know, you can ask me questions later. So what we do is, in the absence of data on what the molecules were doing in the past, we make a simulation. In other words, we model on the computer these molecules and we model the process of evolution. And we see what could have happened. In our model, we have all these proteins I talked to you about, right? These coat proteins, the rads which make buds, the snares which fuse cargo, cytoskeleton which are the tracks, highways, okay? And we model how these little vesicles are formed, how they bud and how they fuse. That's actually very, very simple idea. If you have a compartment and you have stuff on the compartment, you can recruit this budding protein which makes a vesicle, it carries cargo on the vesicle, it moves to another place, it fuses, it checks, have I come to the right place? It checks the address, then it fuses into that, and the system keeps on going. So recruitment, budding, fusion. Here's the mathematics. Mathematically, there's a model of differential equations where we model the size of vesicles, the size of compartments, the number of molecules of various types. Okay? We then model how a vesicle can segregate cargo using various types of biophysical constraints. So if you have a vesicle that's sitting on a compartment, it can choose to take on certain cargo and refuses other types of cargo. We can model that. Once you have a vesicle, we can model how it lands in other compartments. Right? This is mediated by specificity. This is again biochemical details between snares. Right? And I can get into these details later, but I just want to show you the innards of the model. Fine. Then what happens is we can write down what's called a differential equation system, which shows how the different compartments change in size and composition over time. And what we find is actually quite interesting. Okay? If you take a system in our computer and you just put a bunch of lipid compartments and you put a bunch of molecules, these are just molecules that don't know anything about the global structure of the cell, and you run the simulation. What happens eventually is quite beautiful. You end up with a cell which contains compartments of certain sizes with very different compositions. So I think of this as the ER and the Golgi and so on. So in our model, the structure of the cell spontaneously emerges. Okay? More importantly, I'm going to skip through this particular slide. More importantly, you can actually, in our model, evolve the cell where you start from one compartment, go to 2 compartments, 3 compartments, 4 compartments and so on. So in our model, you can actually add compartments by duplicating genes. So this is what the basic hypothesis was. So all we do is we change basic molecular interactions. And what that ends up doing is it changes the morphology of the cell itself. Okay? So this is the cartoon version, right? You have a single compartment, 2 compartments, 3, 4 compartments and so on. And we find that the model itself actually allows different types of compartmentalization. And for those of you who know this kind of language, these compartments are actually steady states of a dynamical system. They're emergent steady states of a dynamical system. And so here's the final idea. You actually have a cell which undergoes various types of processes. Gene duplication, speciation, hybridization, subcellular localization, and eventually a cell that has a few gene copies ends up with many gene copies. This is something that happens all the time. Genes in our bodies constantly duplicate. And the idea is that that's how you end up going from a cell that has 2 compartments to many more. Okay? I'm going to stop there. Just put you back in the context of this picture, which is, I don't know if you can see the reference here, but it's by a person named Bill Martin, who thinks about these kinds of things a lot. There are many hypotheses for how cells arose today. It's very difficult to separate these different hypotheses because we don't have any of the intermediate forms except in the fossil record, and that's very difficult to understand. So one of the ways we are going about it is we're trying to make computer models of these very different procedures to see if there's anything we can learn about whether certain things are possible and other things are not possible. And I still think it's going to be a very interesting field going forward. So, keep your eyes on the news. Let's see what happens. So that's it. I just want to give some credit to my group. These are the students who actually work on the model. And really they are the experts on this, not me. So if you have very difficult questions, I'll ask you to ask them. Thank you very much.